Well, welcome back, everyone, to this afternoon session. It's going to be 3 talks. First one on construction of early embryos in mammals. The second one on construction of embryos in fish. And finally, we will terminate this symposium with a talk on how to keep these embryos running in culture, in vitro. So the first speaker is Nicolas Rivron from Vienna, and he's going to tell us about blastoids.
Thank you, Denis. Thank you so much for bringing us together. What we are interested in the lab is how the mammalian embryo shapes itself in order to implant inside the uterus. And the reason why we are interested in this is because this moment of implantation is, in fact, the bottleneck of human pregnancy. There are many failures that happen at this very specific time point. To give you an idea, about 50% of fertilized eggs fail to develop, and out of those 50%, 75% are failing at this moment of implantation. So this is a critical moment that is, we think, quite interesting to study, and how and why there's such a fragility that happened at this moment of implantation, and how it arises during evolution is absolutely not known, but clearly this is not the case for all mammalians. There are some mammalians, for example the mice, that are very efficient at implanting their embryos. We could imagine that at some point during evolution, this fragility that is specific to human might have played a role to increase fitness by decreasing the number of pregnancies, decreasing the number of progenies. But clearly nowadays, this is leading to all kinds of medical problems, including infertility, early pregnancy loss. And also, this moment of implantation is leading to, is the root of suboptimal fetal development that has very important consequences in adult life. There's a whole field of epidemiology that's called the developmental origin of health and diseases that is quite firm now, and that shows that suboptimal fetal development is actually impacting how you develop cardiovascular disease, for example, 50 years later. And this is all rooted in those first weeks of embryology. So to answer this type of questions, of course, we would need very large numbers of human embryos, which is absolutely impossible. So we came up with an alternative in the shape of stem cells that self-organize into the structures that are quite similar to the pre-implantation mammalian embryo, which is called the blastocyst, and which we called blastoids. And I would argue that this approach will allow us to truly and deeply understand those phenomena of early embryogenesis. So just for the sake of vocabulary, I just wanted to make a couple of explanations. So this is the first fertilized egg. It's going to form a genetically unique cell, and this cell is going to divide into 2, 4, and 8 cells. And during those first moments, there is no lineage segregation, meaning that all the cells are more or less the same. There's fluctuations at protein and transcript levels. However, the cells have equal potential. The system is also very plastic. You can remove one cell and you can still form a full mammalian. And however, when the mammalian embryo switches from 8 cells to 16 cells, then there's a very clear event that happens where the cells on the outside are going to become trophoblasts. These are the green cells that you see here, and these are the cells that are going to form the placenta. And inside, you're going to see a cluster of pluripotent stem cells that are going to form the fetus and also some extra-amniotic tissues. So this is the first lineage segregation. And once this happened, the structure can evolve into our structure of interest, which is called the blastocyst, and which is formed with a fluid-filled cavity, here, surrounded by trophoblasts that is an epithelium that is called the trophectoderm. And inside, we got an inner cell mass. And this inner cell mass very rapidly forms 2 cell types, The first one is those red cells. These are the epiblasts, and they are going to form the full fetus. And those ones are the primitive endoderm cells, and they are going to align here, and these are going to form the yolk sac. And at this point, the whole structure is ready to implant inside the uterus. But what we can do is to harvest it, put it in culture, and to expand the stem cells that it contains. And we can do this with LIF, leukemia inhibitory factor, in order to give rise to those embryonic stem cells. And we can also use FGF4, that was discovered by Denis Duboule in 1998. And in that case, you don't expand the embryonic stem cells, but the trophoblast stem cells. And these are stem cells, of course, you can massively expand them in 2D, you can genetically edit them, you can differentiate them. And they are frozen in time, and the best clue is that you can re-inject them into a blastocyst, and they are going to contribute to the fetal or the placental part. So this was all well established, and so what we did in our case is to combine those two stem cell types and to find the conditions under which they are spontaneously organizing into a structure that is very similar to the blastocyst and that we call blastoids. Now, of course, we can generate very large numbers of those, and I'm not coming back to the technical advantages of such embryo models, but this definitely helps us to better understand the principles undergoing during development. So these are blastoids in real life, and you can see that they are well aligned, and the reason for that is that we actually use those microwells in order to precisely pull a specific number of cells. So we first pull the embryonic stem cells that are going to cluster, and then on top of them we put the trophoblast stem cells, and they are going to coat them. And if we have a specific ratio and specific numbers, especially 8 embryonic stem cells and about 20 trophoblast stem cells, then we have 70% of those microwells that contain a blastoid. And those are free-floating structures. They are not attached, so they're just 3D structures that are deposited on top of the surface. And what you can first observe here is that when you shift away from those absolute and numbers and those ratios, the efficiency is dropping tremendously. So this is first a physical principle that you must have a core of embryonic stem cells that is properly coated by trophoblast stem cells. That's quite obvious that you must come up with specific ratios, but there's also a stoichiometry here. And when we dose those embryonic stem cells inside, we see like all kind of morphometric parameters that are changing. And so these are blastoids that we can pick them up from the microwells, and it was quite exciting at that time because this is in fact a model of the full conceptus. It comprises the epiblast, but also the trophoblast that are going to form the placenta normally, and the primitive endoderm that are actually arising from those cells. In the first protocol, actually the primitive endoderm was underdeveloped. I will come back to this. But because this is like a full structure, we can transfer it back into a uterus. And this is what we did here. And you can see that the uterus is reacting by forming this cocoon that is called the decidua. And this is the normal reaction of the uterus. And of course, we cracked open those decidua after a couple of days. And those cells are proliferating, differentiating, the structure is elongating, but the structure is disorganized. And clearly, nowadays, absolutely nobody has formed a mouse out of a blastoid. A lot of people are trying, but nobody could do it until now. So using those blastoids, we actually want to tackle a very classical question in embryology, which is the one of embryonic induction. In which one group of cells is instructing neighbors. And in the blastocyst, the embryonic inductions have been shown as early as 1978 by a Cope in Oxford. He micro-dissected the left part and the right part, cultured them independently, and saw that those cells are proliferating. So he postulated that there is some mitogenic agent that are coming from here, and it's only 20 years later that Tanaka, in the lab of Janet Ronson, saw that FGF4 is actually produced locally here, and that this is regulating proliferation of the trophoblast. So this is where things were standing since, in fact, 1998. So kind of like re-explored this concept of embryonic induction by systematically comparing blastoids with trophospheres. And trophospheres are formed in exactly the same way as blastoids, but without the inner cells, without the embryonic stem cells. And you can already see that those trophospheres, you can see one here, the second one here, and the third here. You can see that the trophoblasts are quite different from the trophoblasts from blastoids. And so clearly, we already have clues about what those embryonic inductions are doing. So the first thing that we do, of course, is to check the original hypothesis from Cope and Tanaka. And indeed, in the presence of the embryonic cells, the number of trophoblasts is increasing, so they are proliferating. In their absence, they don't. So clearly, those cells are inducing the proliferation of the trophoblast. And a second function that we looked at is self-renewal. So we can pick like single trophoblasts from blastoids and trophospheres, put them in 2D, and see if they can form a colony. And this is what we measure here. This is a logarithmic scale, and you can see that the trophoblasts from blastoids have a much higher propensity to form a colony as compared to the trophoblasts of trophospheres. So this is self-renewal. And when you have, of course, when you have self-renewal and proliferation, these are the two cardinal functions of stem cells. So what those red cells, what those epiblast cells are doing is that they are maintaining the stem cells that are going to form the placenta. So now we can go into the molecular mechanisms, and this is, we started by doing single cell sequencing. So these are like trophoblasts that were isolated from blastoids here, and trophoblasts isolated from trophospheres. And you can see that there's a shift between the two. This is a control. These are trophoblast stem cells cultured in 2D. These are the embryonic stem cells. And you can see clearly that there's a shift of transcriptome between the trophospheres and the trophoblasts of blastoids, which we use to infer the molecular regulations that are due to the inductions. And I'll just show you like this table, but beyond FGF4 that was already described, we found that those epiblast cells are producing LPA, which is a GPCR ligand regulating the Hippo pathway. It's also regulating, it's also producing IL-6, a lot of Wnt, nodal, BMP4, BMP7. So you see that there's a whole world, there's a whole number of molecules that are actually like being secreted by those cells. And then we can look at the pathways that are activated into the trophoblast. And we can see the MAP kinase pathway, very interesting because, of course, this is related to FGF4. But you can also see the Hippo pathway, and we can also see the JAK-STAT pathway, the Wnt pathway, and the TGF-beta pathway. So it looks like, at least at the molecular level, there's a lot more than FGF4. And of course, it was already known for quite some time that there are signals, the signals coming from here, whatever they are locally regulating those cells, and that those cells are becoming a little bit distanced, and they can differentiate. And you can look at this by looking at CDX2. This is a homeobox gene. It's very nicely expressed everywhere into the early blastocyst. And then when the blastocyst is growing bigger, you can see that CDX2 is very high on one side, which is on this side, and it gets lower on the other side. And on the other side, there are differentiation markers, including, for example, keratin 18. And so now that we have like more molecules, we want to know actually what they are doing. And in order to look at one function, we did this assay where we are plating trophoblast stem cells with a CDX2 EGFP reporter. So that's a knock-in inside the CDX2 loci. And then we can put one by one those molecules, and we can put them in combinations, and then we look if this is regulating CDX2. And we actually did this for combination of all kind of combination of those molecules. And what we found that was quite surprising to me is that there is only one combination that was capable of maintaining a very high CDX2 in the trophoblast stem cells. If you use different combinations of the same factors, you actually fall back into a state that is very similar to the classical trophoblast stem cells CDX2 levels. And we found actually many combinations for which the stem cells cannot be maintained. So these are the passages in 2D. We cultured the cells for 10 passages with those specific combinations. And so those are probably inducing differentiation, and there's a very precise combination that maintains very high levels of CDX2. And those cells, now we can analyze them, and they are very enriched for the trophectoderm genes. And now when we plot them along pseudotimes, we actually call them trophectoderm stem cells instead of trophoblast stem cells. You can see that along a pseudotime, the trophoblast stem cells, the classical one from Janet, are very nice cells, but they comprise multiple subpopulations, including, for example, this one that is expressing DPPA1 and ASCL2. And these are actually post-implantation genes. These are genes from the ectoplacental cone. So it looks like those trophoblast stem cells, they are comprising some progenitors, probably the blastocyst-like, but they also comprise other subpopulations that are post-implantation. And now what we do, if we expose the trophoblast stem cells to a very precise combination of molecules coming from the epiblast, we maintain those trophectoderm stem cells that start expressing a new set of genes, including KLF5 and TAD4. This was quite interesting for us because these are the two knockouts. When you erase that gene, you cannot form a trophectoderm. And to my knowledge, these are the only two ones. And of course, the pseudotime, you can see, is like lower as compared to trophoblast stem cells as compared to differentiated stem cells. So this is pointing at the fact that those cells are a little bit earlier in time and that they start expressing other type of genes. And we can also look at their colony formation efficiency. It's much higher as compared to, 2 times higher as compared to trophoblast stem cells. We can derive trophoblast at a much higher rate using this new cocktail as compared to the classical cocktail, which support the idea that those molecules are active during the blastocyst stage. And we are actually preventing the formation of those differentiated cells, those keratin 8 cells. So we have about like 15% normally in the trophoblast stem cells, and we get pretty much 0% in the trophectoderm stem cells. So those molecules are here to maintain very high levels of self-renewal locally and to prevent the differentiation of the trophoblast. And of course, when you think about the embryo, the idea is that the embryo is using this optimal set of factors in order to maintain very high self-renewal in this region, and that when the embryo is changing geometry and changing shape, those factors that are coming from here are going to maintain very locally a stem cell niche in this region, and as soon as you get away from it, you fall into a suboptimal state that allows you to differentiate. And this is, of course, very important because you must form all those differentiated cells that are supporting the embryo. So those factors are clearly regulating the growth. FGF4 was already known. We add up on top of this. And of course, this is to maintain the stem cells here. And those stem cells are the ones that are going to form the full placenta. And we came up, along the way, we actually came up with a second function for those embryonic inducers that we had not seen before. And I'd just like to present it to you here. So when we put the blastoids into the uterus, I told you that they form those cocoons, those decidua. Just for as a sanity check, we actually fixed those blastoids before we put them into the uterus. They are incapable, those dead blastoids are incapable of inducing decidua. Telling this because it was very controversial. You know? But I think that this experiment is proving that there's an active process of the blastoid or the blastocyst instructing the uterus to form the cocoon. You know? If this is time for me to implant, please form a cocoon around me. You know? And this is confirmed by this experiment when we actually can transfer blastoids or trophospheres. Those trophospheres, they are more differentiated, they proliferate less, they self-renew less, and they are incapable of instructing the uterus. So they can actually stick, they're actually very sticky. They can stick to the uterus, but they cannot tell the uterus to form the cocoon. So this experiment is suggesting that, in fact, it's those cells that would be instructing the uterus. And what we can do, because we know that, because now we have those trophectoderm stem cells, we can also do the transfer, and you can see that this is blastocyst using the same procedure. About 40% of the blastocysts are forming a decidua. In our original protocol, it was 8% for the blastoids, and now when we have trophectoderm stem cells that have higher CDX2 levels, we have 20%. So it looks like this is improving, and now what we can do is to only regulate CDX2, this homeobox gene, and when we dox those blastoids, we can also, those blastoids acquire a much higher competency to instruct the uterus. So it looks like this instruction is going through CDX2. And so we looked into what CDX2 binds to, and we actually found that it binds to many things, but among them Wnt7b and Wnt6 that are clearly present in the trophectoderm and in post-implantation trophoblast, and that when we knock out Wnt7b and Wnt6 only in the trophoblast and form the blastoids, those blastoids have a lower capacity to form the decidua. Here we are measuring the diameter of these decidua. We are dropping by 10 to 20%. Just a small reminder, this means like that the volume of the sphere is divided by 2. So that's actually very significant. So we came across this second function that is, we don't know which one is regulating it, but it goes through CDX2, and that secretes Wnt6 and Wnt7b that is instructing the uterus to form the cocoon. So this is the second function, and we actually came across a third function, which is this one. So here we are measuring the diameter of the blastoid or the trophosphere, and what we see is, so this is the diameter of a blastoid, and this is the diameter of a trophosphere. It's smaller, and then what we can do is to replace the embryonic stem cells by the factors that they produce. And we can dose BMP4 and nodal on top of those trophospheres. And when we measure the diameter, you can see that the diameter is increasing for BMP4 very clearly, very significantly also for nodal. And the two together, we almost get a rescue. And so the first thing that we did is to count the number of cells. The number of cells is absolutely flat. So those factors are not regulating proliferation, but we can do a knockout only in embryonic stem cells, and you can clearly see that you're decreasing the size of the cavity. And we found out that it's regulating CDX2 and KLF6, tight junctions and epithelial junctions. So we think that nodal is actually regulating those tight junctions to seal the trophectoderm. And we can knock out KLF6 only in the trophoblast stem cells. We get a small cavity also. So we're coming to the model that, in fact, those factors, you can see that this is a wild-type trophosphere. We can increase the size with those two factors. Trophospheres with KLF6 knockout are smaller, and they are incapable of responding to it. So we think that overall, those inductions are also here to regulate the epithelial morphogenesis, to seal this epithelium and allow it to pump water so that it can swell like a balloon. And this is very important, of course, because by swelling, you actually distance those cells from the embryonic inducers, and those cells can now differentiate and become sticky. And these are the sticky cells that are attaching to the uterus. So this is the first axis that appears, that is extremely important to maintain the stem cells on one side and attach to the uterus on the other side. This is a beautiful picture taken by Anne Sutherland, and you can see that this is a blastocyst that is implanting into the uterus via the opposite side as compared to the epiblast, those sticky cells. So overall, what we understand is that those extraembryonic tissues and organs that we all know of are very important for later development in order to nurture the growth of the fetus. But what we and others are thinking now is that at the very early stage, it's actually the opposite. It's those epiblast cells that are putting a lot of effort into instructing the development of those cells here. And it's doing so in order to invest on its future by making the sticky cells here to implant inside the uterus and the stem cell of the placenta in order to form the structure. So of course we got very interested into whether the same principle is happening in human. And one reason why we got so interested in this is because I told you that the sticky cells of the mouse are on this side. It turns out that the sticky cells of the human are exactly on the other side. It's very strange, but during evolution there's a 180 degrees flip of the axis, and now human embryos started to implant via this side. And you can imagine that this might have had very important consequences on how the embryo is implanting and how this is interacting with the uterus and how the whole thing is taking shape afterward. So in order to do this, we did human blastoids, and we used naive human embryonic stem cells. These were developed by Austin Smith. It's the PXGL culture. And you can see that when we triply inhibit the ERK, TGF-beta, and Hippo pathway, the cells know what to do, and they actually form blastoids very efficiently. We did it with 5 cell lines, and we got more than 80% of the microwells that are forming those structures. And those structures are very nicely organized. You can see that they got quite tight numbers and quite tight ratios of cells. We get the epiblast cells, the trophoblast cells, and the primitive endoderm cells. And we can check by single cell sequencing what those cells are. So these are the naive cells to start with in 2D. And then you can see that between 24 and 60 hours, those cells are forming 2 cell types. The first one is transcriptionally very close to the naive embryonic stem cells, and this is the epiblast of the blastoid. And the second one is the trophoblast. And only in the second step, between 60 and 96 hours, we got the third cell type that appears. And these are the primitive endoderm cells. And at the end, we have only 3 cell types. And now we have to project those cells onto reference maps to understand if we have formed the right cells. And we did the experiment, but I'll just show you the results of an international consortium that checked our results. You can see that this is the blastocyst. It starts at E5, E6, E7. This is the trophectoderm, epiblast, primitive endoderm. You can see that the cells are falling very nicely onto the trophectoderm, epiblast, and primitive endoderm. We have about like 2 to 3% of the cells that go off target. So of course, beyond the molecular signatures, what we want to know if those structures are functional, and of course, it is actually unethical and now absolutely forbidden by an International Society for Stem Cell Research to transfer human blastoids into any type of uterus. But what we can do is to do an implantation assay in vitro. And so in order to do this, we culture human endometrial organoids. This is the lining of the uterus. And then we can dissociate the cells, put them in 2D in order to facilitate the deposition of the blastoids. And that was our second nice surprise, because when we actually put the blastoids on top of those layers, they actually do nothing. You can leave them for 2 days and absolutely nothing is happening as far as we can see. But if we have stimulated the endometrial cells with progesterone and estradiol, which are the hormones of pregnancy, those cells are changing, and then they become capable of interacting with the blastoids. And you can see that 30 to 40% of the blastoids are attaching, and they start to grow. And they don't grow in a chaotic way. They actually start to grow from the polar side, which is like the beginning of placentation. And so, of course, this led us to do some live imaging. And we actually observed that all the blastoids are attaching via what is known to be the sticky side, the polar side of the blastoid. This is not anecdotal at all. It's actually very efficient. You can see here on those movies that we are playing with the pipette of blastoids that just attached. And you can see that they are like very firmly attached via always the same pole. So this tells us, of course, that the blastoids are formed an axis spontaneously and somehow. And we actually looked at NR2F2, which is a marker that was found by Laurent David in Nantes. And you can see that NR2F2, which is very nicely expressed in the sticky region. And those are the cells that are in close proximity to the epiblast. And so this is suggesting that those epiblasts are inducing the local maturation of those cells. And if this is the case, if we manage to get rid of those inner cells, we should see a phenotype. And this is what we did here. We actually inhibited the STAT pathway or overactivated the hippo pathway, the hippo pathway. And in both cases, we eliminate the cells in the center and we form trophospheres. Those trophospheres are incapable of interacting with hormonally stimulated endometrial cells. So it fits with the idea that those epiblast cells are instructing locally the cells to mature and become sticky. And we actually had another control. These are post-implantation human trophoblasts, and we can form aggregates. They are also incapable of attaching. So when you didn't form your mature domain, you cannot attach, and when you're too late, it's too bad. So, and we talked about it to clinicians, and of course they told us, we know that for 50 years. This is called the window of implantation. So we think that we are recapitulating some aspect of this window of implantation in vitro. So now, of course, we can extract the markers here and make some pair of ligand receptors, and we have a whole list that can be interesting. We can also culture those blastoids further. We did 6 days after day 7, which is the developmental stage of a blastoid, so day 13, and we find those structures. So clearly these are not day 13 alike, but the cells are growing and they are differentiating. So now we are in the position of trying to understand how this axis is forming in the mouse and in the human, how things have evolved from one to the other, and what's the impact on the implantation. I'd like to finish by saying that so we put a lot of effort into regulating this type of research, and I think we have to admit that blastoids are quite good models, but they are also very rudimentary, and they're also very different from embryos. So these are clearly not embryos. And there was an update of the ISSCR guidelines that is proposing some ethical oversights for this type of research. I'm very happy to talk about it. If you're interested, you can look at those ethical papers that we participated to. And I'd like to thank the team, especially Giovanni, who is doing all the single-cell sequencing analysis. Jinu, who did the mouse trophectoderm stem cell part, and that will come out in the next few weeks. And then we have Alok, who did the human blastoid along with Haru and with Haydar. And Haydar put actually the effort into the implantation assay. And that's it. Thank you very much.